Hello everyone, my name is Anubhuti Pandey and today we will learn about cell organelles. So let's begin with our first cell organelle, that is endoplasmic reticulum. So what is the meaning of endoplasmic reticulum? When we look at endoplasmic reticulum with the help of electron microscope, we see that they look like a network of reticulum. They are made up of tiny tubular structures and they are scattered in the cytoplasm. Hence the name endoplasmic reticulum. They are made up of three things. First one is cisternae, second is vesicle, and third one is tubules. So what are cisternae? As you can see here in the diagram, this is cisternae. Cisternae, they are long and they are flat. As you can see here, they are flattened structure and they are unbranched units, and they are also arranged in stack. They are arranged in stack one by one. Next one is vesicle. As you can see here in the diagram, these are vesicles. Vesicles, they are oval and they are membrane bound structures. Next one is tubules. This one is tubule. Tubules, they are irregular and they are branched. As you can see here, they are branched structures and they are bounded by membranes. Tubules are free or associated with cisternae. They either exist freely or they are associated with cisternae. Now let's look at the types of endoplasmic reticulum. So there are basically two types of endoplasmic reticulum. First one is rough and second one is smooth. So what is the difference between rough endoplasmic reticulum and smooth endoplasmic reticulum? Rough endoplasmic reticulum are granular structures. They are rough structures, and smooth endoplasmic reticulum are agranular because they do not contain any granules. So what do we mean by granules? Granules means the presence of ribosomes. So rough endoplasmic reticulum have 80S ribosomes, which is bound by their larger subunit, and they are binded, as you can see here in the diagram. Ribosomes are binded with the membrane of rough endoplasmic reticulum with the help of two glycoproteins. These glycoproteins are ribofourin 1 and 2, and they are present on the surface of rough ER. When we look at the smooth endoplasmic reticulum, as you can see here in the diagram, there is no ribosomes or ribofourin present. Rough endoplasmic reticulum is mainly made up of cisternae. These are cisternae, and smooth endoplasmic reticulum are made up of tubules. So where rough endoplasmic, what is the function of rough endoplasmic reticulum? So it is used in the protein synthesis and secretion, and it is present mainly in the cells of liver, pancreas and goblet cells. And smooth endoplasmic reticulum are concerned with glycogen and lipid metabolism. They, in animal cells, they are present in the they are used in the synthesis of steroidal hormones, and they are present in adipose tissue, interstitial cells, muscles, glycogen storing liver cells and adrenal cortex. Now let's look at the functions of endoplasmic reticulum. So they perform various functions. First function is mechanical support. So microfilament, microtubules and endoplasmic reticulum, they together form the endoskeleton of the cell. They form the skeleton of the cell, just like we have skeletal system in our body. Intracellular exchange. So whatever that is synthesized in this cell is exchanged with the cytoplasm. That is, transport of material in cytoplasm from one place to another may occur through the ER. What is the function of rough ER? Rough ER is actively involved in protein synthesis, and smooth ER is actively involved in lipid synthesis. They are also helped in the synthesis of lipoproteins, means lipid and proteins, and they are involved in the synthesis of glycogen. Next is cellular metabolism. So reticulum provide an increased surface for metabolic activities within the cytoplasm. So whatever the metabolic activities takes place in the surface of surface of endoplasmic reticulum. Next one is formation of nuclear membrane. Whenever the nuclear membrane gets disintegrated, ER helps in the formation of nuclear membrane. New nuclear membrane is formed during cell division with the help of ER. Next is formation of lysosomes. So many organelles are formed by ER such as lysosomes, Golgi body and microbodies. They are also helps in the detoxification, especially smooth ER is involved in the detoxification of drugs, pollutants and steroids. Cytochrome P450 in ER act as an enzyme which function in detoxification of drugs and other toxins. So cytochrome P450 that is present in the ER is actively involved in the detoxification. And the last function is precursor of secretory material in Golgi body. Whatever that is synthesized is then secreted in the Golgi body. So this was all about endoplasmic reticulum. If you like this video, please like, share and subscribe to Spindle Classes. Thank you so much.